How do fragile pharmaceutical products remain stable for years without liquid water? The answer lies in one of the most sophisticated processes in pharmaceutical manufacturing, lyophilization. Lyophilization is a process used to remove water from pharmaceutical products. By reducing moisture, it helps improve stability and extend shelf life. Today, it is widely used across the pharmaceutical industry. Many pharmaceutical products are unstable in liquid form. Over time, moisture and chemical reactions can affect product quality. This is especially important for biologics, vaccines, proteins, and injectable therapies. To improve long-term stability, water must often be removed. Pharmaceutical lyophilization consists of three main stages: freezing, primary drying, and secondary drying. Each stage plays a critical role in producing a stable final product. The core of lyophilization occurs during primary drying. Here, frozen ice is removed through sublimation. Instead of melting into liquid water, ice changes directly into vapor. This allows water to be removed while preserving product structure. Although the concept sounds simple, the process is highly controlled. Product temperature, chamber pressure, heat transfer, and formulation behavior all must remain within carefully defined limits. If process conditions are not properly controlled, defects can occur. Products may collapse, crack, retain excess moisture, or lose critical quality attributes. Successful lyophilization requires precise process design. Today, lyophilization supports a wide range of pharmaceutical products, including vaccines, biologics, injectable medicines, and advanced drug therapies. It remains one of the most important preservation technologies in modern pharmaceuticals. This was a brief introduction to pharmaceutical lyophilization. In the next video, we'll explore why pharmaceutical products require lyophilization in the first place, and the stability challenges that make freeze drying essential. Thank you for watching.